Okay, moving on to objective 3 here, we're thinking about what's happening as far as the response on the postsynaptic cell. And we're going to speak about 2 types of responses here. One is a fast response and one is a slow response. And we kind of introduced that idea when we drew those synaptic events out. So basically when that ligand, which is our neurotransmitter, binds to a ligand gated channel on the postsynaptic side, it's going to trigger some type of response in that cell. The distinction here is within the speed with which that response takes place. If it happens really quickly, we're going to call that a fast response. And we'll talk about the details that allow for that fast or rapid response. If it takes more time, if it's more slow or more gradual, we'll talk about what happens for that slow response as well. So basically, we've got these channel-linked receptors that are called ionotropic receptors. And ionotropic receptors are basically where the receptor is also acting as a channel. This is what constitutes that fast response. So basically just introducing the response that happens really rapidly or really quickly. And the reason it happens very rapidly is because the receptor is also a channel. It's kind of like a 2-for-1 scenario here. So when the ligand binds to this receptor, also acting as a channel, it's going to open up that channel and it's going to initiate ion movement. Ion movement, we know, is synonymous with membrane potential change. In other words, it could lead us to a threshold change, which could create an action potential on our postsynaptic neuron, okay? And basically, we're going to have the closure of this channel when that neurotransmitter leaves or unbinds from that receptor, okay? So essentially, just to kind of summarize this here again, we've got the fast response associated with an ionotropic receptor, and an ionotropic receptor is synonymous with an ion channel, right? It's the same structure having two actions or 2 functions in this membrane. So when that ligand, which is our neurotransmitter that crossed the synaptic cleft, binds to this receptor, it's going to trigger the opening up of this ion channel. Ions are going to move according to their ECDF, and then we're going to get closer to depolarizing this membrane if we have a threshold stimulus. So that was the fast response, right? And the reason it's fast, just to make sure that you're making the connection here, the reason it's called a fast response is because everything is happening in one spot. The receptor is also a channel. So as soon as it binds that neurotransmitter, we can immediately start to see ion movement. We can immediately start to see cellular response. That can be compared to a slow response. And a slow cellular response is associated with a G protein coupled receptor, also known as a metabotropic receptor, which is much more slow because the receptor here is only the first step in the process, okay? This is where we begin to speak about what's called signal transduction. So we can think of the metabotropic receptor as being sort of a middleman in a long series of events that eventually leads us to a cellular response. It's not as rapid as what we saw with the ionotropic receptors. And so when our neurotransmitter binds, right, crosses the cleft, right? Think about that connection there from the synaptic events, binds onto our receptor in the cell membrane on the postsynaptic side. It's not going to be the end of the signal. It's going to have some other steps that are involved before we can see that cellular response. So it's going to actually create a conformational change in the protein that is associated with this receptor, right? This is a receptor that is coupled to a G protein. So here's that G protein with 3 different subunits here in blue. And so the binding of our neurotransmitter triggers a configurational change in our G protein such that one of the subunits, specifically the alpha subunit, dissociates off, floats downstream in the membrane, and binds to another enzyme. This is called our amplifier enzyme. The amplifier enzyme will then generate what's called second messenger. We have different types of second messenger, but it is usually cyclic AMP or sometimes cyclic GMP, which again is one of our nucleotide-derived molecules, okay? So we have the alpha subunit of our G protein activating this amplifier enzyme, and we'll put in some names here as we go along. But for now, we're just going to think of it as an amplifier enzyme, and that amplifier enzyme makes our second messenger, and then that second messenger goes and incites or initiates the cellular response. So I say all of this to say that it's really this passing of the baton, kind of like a relay race, right? One structure takes care of one part of this signal transmission. Then we have the subunit dissociating off. Then we have the amplifier enzyme. Then we have some second messenger, right? So a lot of steps that are involved in the transmission of this signal. And so it's not as quick or as rapid as what we see with the ionotropic receptor, which has a more of a fast response, okay? Now, we'll talk a lot more about the details of what this second messenger does in order to generate the cellular response. But the second messenger usually opens up some type of channel, right? So the receptor here is not the channel, okay? The receptor is what starts the signal, but the channel is where we ultimately see the initiation of the cellular response. And then there are some steps in between. Another model of how this slow response takes place is called direct coupling. So not necessarily involving any second messenger as we see in this first model, but in the second model here, we have the neurotransmitter binding to our metabotropic receptor, the receptor being coupled or associated with or connected to a G protein. We then see a configurational change in that G protein such that the alpha subunit dissociates off, moves down in the membrane, and instead of coming in contact with an amplifier enzyme or other steps in the process, it directly opens up an ion channel, and then we can see ion movement, which will then generate that actual cellular response, okay? So two different models here for the slow response, but they both have the same general idea. There are some steps involved in the process. The receptor is only the beginning of that signal transduction, and then we have to pass that signal from one structure to another structure, and then we can open up an ion channel, and then we can see cellular response, okay? All right, I'll pause here and see how we're doing with that and what questions we may have about that, and then we'll speak about what those different ion movements would look like. Okay, so when we think about graded potentials, this is kind of where they come into play. So we spoke a lot about graded potentials, and we should be making the connection that graded potentials occur or are initiated along the soma and the dendrites, right? The proximal parts of the neuron. And so when we go from a presynaptic neuron to a postsynaptic neuron, the neurotransmitter that crosses the junction is actually directly involved in creating a graded potential, okay? So although we spoke about them previously, I really want you to think about how they connect to what we're speaking about now, right? And where they fit into the broad scheme of things. So we open up a ligand-gated ion channel, and this allows ion movement to flow. And this could change the membrane potential, right? If we have positively charged ions or cations moving into the cell, that's going to depolarize the cell. If we have positively charged ions or cations moving out of the cell, that is going to hyperpolarize the cell. And so based upon the specific ion that we're moving and the direction that it is moving in, we could depolarize the membrane, right? Or we could hyperpolarize the membrane. And this is what we mean by graded potentials being able to get us closer to an action potential. So we revisit that exact same idea that graded potentials can be summed, and they could contribute to getting us closer to threshold, and then we can fire another action potential on this postsynaptic neuron. Now, if we open up a sodium channel or a calcium channel based upon the ECDF of these two ions, this would result in a depolarization. This is called an excitatory postsynaptic potential, or an EPSP. All right, if we had enough of these types of events, we would definitely get to threshold. We would definitely fire another action potential on the adjacent neuron. On the other hand, if we opened up a potassium channel or a chloride channel, this would result in a hyperpolarizing event, okay? Remember, potassium has an outward ECDF. Based upon all of the events that we described previously, if we open up a potassium channel, positive charge is going to leave the cell, the membrane potential is going to become more negative. This actually makes it harder for us to get to threshold, and this is called an inhibitory postsynaptic potential, or an IPSP, all right? Same thing with chloride. Chloride has, depends on the type of cell. So cells that have active transport of chloride have an outward ECDF, but cells that have no active transport, and we'll clarify this going forward, they actually allow chloride to stabilize the membrane potential. So not necessarily make it more positive or negative, but negate any positive membrane potential change and just keep it stable at rest. All right, and we'll add some more clarity in terms of chloride as we go along. But the main take-home message here is that a greater potential can be initiated depending on what type of ion channel we open up and what direction of ion movement we see, which would either lead to an EPSP, which excites the membrane, right? It depolarizes the membrane, or an IPSP, which does the opposite. Okay. So again, just reiterating that idea here, different neurotransmitters are going to result in different types of potentials. So the most common neurotransmitter involved in EPSPs is glutamate. Glutamate is usually referred to as an excitatory neurotransmitter. It's going to help depolarize cells and cause excitation on neurons. And the opposite neurotransmitter is GABA. And GABA is often associated with inhibitory postsynaptic potentials, and those basically stabilize or hyperpolarize the membrane potential, okay? So just to make an association with some neurotransmitters and the types of membrane potential we typically see on our neurons when these neurotransmitters are released. Okay, so again, adding some clarity here in terms of that excitatory postsynaptic potential versus that inhibitory postsynaptic potential. So excitatory potentials make the membrane more positive. It depolarizes the membrane, and it's going to make it more likely or more easy for an action potential to be generated. It's going to make it, it's going to get the membrane potential closer to that threshold, okay? Whereas inhibitory postsynaptic potentials do the opposite. They make it harder to fire an action potential. They take the membrane potential away from rest, but in a negative direction. They hyperpolarize the membrane potential, and it's going to require a stronger stimulus to overcome or overcompensate an inhibitory postsynaptic potential in order to get us to an action potential, all right? They also have the ability to stabilize the membrane potential. This is really relevant specifically to chloride, and I'll talk about how chloride can actually not depolarize or hyperpolarize, but just keep the membrane potential stable. All right, I'll pause here again briefly and see how we're doing with that. What questions we have about this so far. So just to kind of recap and summarize here, we're speaking about fast responses versus slow responses. Fast responses open up a channel quickly because the receptor is also acting as a channel. Slow responses open up a channel, but it's really the final step in a sequence of events that involve several different players, okay? Whenever we open up that channel, depending on the type of ion that is being moved, we can then generate an excitatory postsynaptic potential, which depolarizes the membrane potential and gets us closer to threshold, right? Closer to an action potential. Or we could hyperpolarize the membrane, which takes us further away from threshold, makes it harder to fire an action potential, or in the case of chloride specifically, we can stabilize the membrane potential. Okay, so that's kind of what we've been describing so far, and that kind of is summarized on this slide here as well. So we're comparing the events of that fast response to the events of the slow response. So the fast response involves an ionotropic receptor, which is also acting as an ion channel. So when the neurotransmitter binds to this ionotropic receptor, it opens up an ion channel that allows sodium to rush in, potassium to rush out, and because of the overwhelming inward flow of sodium with respect to potassium, we depolarize this membrane and we make it easier to fire an action potential because this constitutes an EPSP, an excitatory postsynaptic potential, okay? If we think about our slow responses here, we said the neurotransmitter binds to the receptor. The G protein that is coupled to that receptor has a change in its conformation such that the alpha subunit floats away, moves downstream in the membrane, activates this amplifier enzyme, right, which is adenylate cyclase in most cases, but it can be other things, which we'll talk about later on. That will then generate, so the amplifier enzyme then generates or produces second messenger, which is usually cyclic AMP. And then cyclic AMP, it will then create the opening of the channel. Now, depending on what type of channel we open up, that will determine if it's an EPSP or an IPSP. If we open up a sodium channel, sodium is going to rush in, that's going to be an EPSP. If we open up a calcium channel, calcium will rush in, that will be an EPSP. If we open up a potassium channel, potassium rushes out, that will be an IPSP, okay? Now I want you to think about what would happen if we close these channels as well. So opening up a sodium channel allows sodium to rush in and depolarizes the membrane. But if I were to close a sodium channel, I would prevent sodium's movement, and that would be the opposite type of event. That would be an IPSP, okay? Let me say that again. If I open up a sodium channel, sodium can move, that generates an EPSP. But if I close a sodium channel, sodium cannot move, that would constitute an IPSP. Same thing for potassium. Open up a potassium channel, potassium can rush out, that generates an IPSP, but if I close a potassium channel, potassium cannot move, and that will generate an EPSP, okay? So we want to think about these in the direction of opening the channel and ions being able to move, but also think about them in the direction of closing the channel and that ion not being able to move and what that would do to the membrane potential if we didn't allow that ion to move. Okay.
I have a small question.
Yeah, sure.
I don't know if it's entirely related, but how are millivolts measured if a neuron is microscopic?
So that's a good question, and this is all pretty abstract. So this is kind of theoretically how we would measure that voltage.
Oh, okay.
But no one has ever stuck a voltmeter right into a neuron. It's sort of theoretical in terms of the direction of the membrane potential change. But that is a really good question.
Okay.
Yeah, so that's just kind of how we represent this in terms of what we think happens inside the neuron. But no one has actually ever seen this or physically measured this.
Okay, so it's more just symbolic of what we think happens.
Right, yes, perfect. Okay, really good question. And if you have other questions like that, please feel free to ask them because a lot of this is easier to understand when you understand the context of how this is being presented. Okay, so thinking about our inhibitory synapses here, we're looking at chloride. Did we skip a slide? Okay, no. Yeah, so we're thinking about potassium leaving the cell or chloride channels being opened up. And those two types of events could constitute an IPSP. If we think about potassium moving according to its ECDF, moving out of the cell, that is an inhibitory postsynaptic potential. Whereas if chloride channels open up, that could be an inhibitory postsynaptic potential, but it can also have a stabilizing effect on the membrane potential. It really depends on whether or not we have active transport of chloride on that specific cell. Okay, and just looking at the example here, in this fast response, we open up this ion channel, potassium is allowed to leave the cell, that is an IPSP. Okay, positive charge leaving the cell, that constitutes an IPSP. And it's fast because the neurotransmitter binds to a receptor that is also acting as that channel. If we think about neurons that does have the ability to actively transport chloride outside of the cell. So we're pumping chloride outside of the cell. We now have a gradient where chloride will move into the cell according to its ECDF if we were to open up these chloride channels. And so if that were to happen, chloride would move into the cell, that would constitute an IPSP. So on neurons or cells in general, we can think about muscle cells as well, on cells that have active transport of chloride, right? Chloride being pumped out of the cell, if we open up a chloride channel, that is an IPSP, okay? On cells that do not have active transport of chloride, okay, we don't have that inward ECDF. And so if we were to, we don't have that active pumping of chloride out of the cell. And so this is what it would look like at equilibrium. We would have free movement of chloride into and out of the cell being matched. But if we were to open up a sodium channel and we had this chloride channel also open up, it would basically cancel out any positive ion movement that is coming inside of the cell. So just kind of reiterating this here. If I open up a chloride channel on a cell that is being depolarized, every time a positive charge comes into the cell, a negative charge comes in as well, right? For every positive sodium that comes in, a negative chloride comes in. And this is how chloride can stabilize the membrane potential on cells that do not have active movement or active pumping of chloride on the membrane, okay? So just to kind of summarize here again, if we have active transport of chloride on this neuron, then opening up a chloride channel causes an IPSP, right? If we don't have active pumping of chloride on this channel, then opening up a chloride channel just cancels out any positive ion movement. So for every positively charged ion that comes in, chloride comes in and cancels out that membrane potential change. And this is how we can stabilize the membrane potential on cells where there is not active transport. Okay. And just looking here at the events of how IPSPs or these graded potentials can lead to cellular response. So if we have a higher frequency of action potentials, we have more neurotransmitter being released from our presynaptic neuron. We have more of that neurotransmitter crossing the junction, crossing the cleft, binding to those receptors, either opening them, right, or closing them, respectively. And we have a greater, or we can think about it in either direction, either more of an influx or less of an influx of the ion permeability. That would either increase ion movement or decrease ion movement, and that would lead us to a greater hyperpolarization on our postsynaptic cell, okay? And the reason that we're thinking about this in the positive or negative direction is because let's say we opened up a potassium channel that would allow potassium to leave the cell, that would increase ion permeability, right? More potassium moving, which would cause greater ion flux. That would lead to a hyperpolarization. But as I mentioned earlier, we can also think about some of our positively charged ions and what would happen if we closed those channels. So if I were to close a sodium channel or close a calcium channel, that would cause a decrease in the ion's permeability, which means less of that ion being moved, right? Less ion flux, but that would also constitute a greater hyperpolarization or an inhibitory postsynaptic potential. Okay, so we can think about the opening up of the channel leading to an IPSP, right? Or the closing of the opposite types of channels also leading to an IPSP. And then the last thing I want to point out on this slide is really just looking at the frequency here. So this is how we talk about different types of stimuli. And we made this point before. In the nervous system, we cannot change the amplitude or the duration of action potentials. And so the way that we can send a signal, the way that we can communicate greater signals is by manipulating the frequency of the action potentials. So higher frequency of action potentials, right? If you're firing more action potentials per time, that relates or translates to more neurotransmitter being released, and that relates or translates to more of a synaptic signal, which can be read or transmitted on the adjacent neuron.